There are 3 domains of life on Earth. We have our bacteria, sometimes called eubacteria, archaea, and eukarya. Because these 2 domains, bacteria and archaea, are so similar, we often colloquially refer to these 2 as prokaryotes, and often talk about them in comparison to eukaryotes. Prokaryotes, as you previously learned, are what ruled the Earth for over 1 billion years. But we believe that through endosymbiosis, eukaryotes eventually arose from prokaryotes. Prokaryotes are simple in structure. Building from what we discussed before, prokaryotes meet all 5 of our key criteria. So yes, they are indeed cells, they're just very simple cells. They have genetic material, such as DNA, which is found in this region shown here, but also they have something special that is unique to prokaryotes in that they also have small circular DNA floating around inside of them called plasmids that are separate from their chromosomal DNA and help provide survival advantages, such as antibiotic resistance. Our prokaryotes also have ribosomes, as well as cytoplasm. They have a barrier separating the cell from its surroundings, their plasma membrane, and they have a means of making and using energy. They have metabolism. Now, in addition to these structures, you will notice that a prokaryote also often has something called a capsule, as well as a cell wall. These structures just provide extra support for our prokaryotic friend, separating them from their environment. These are much more rigid structures. And then they also have pili and flagellum. The pili help increase surface area on the prokaryote, which helps them with transport of materials and interacting with their environment. And then the flagellum helps propel the single-celled prokaryote through its surroundings. So that's largely for locomotion. A prokaryote, though, does not have any membrane-bound organelles. What you'll notice here is that there is a region within the prokaryote that is highlighted as the nucleoid region. So the DNA, or genetic material that resides within a prokaryote, is not within its own organelle. It is kind of free-floating, but it's free-floating within a specific area. And that area we call the nucleoid, or the nucleoid region.
So now, for your next check-in question, go ahead and label the following structure on our prokaryotic cell. Keeping in mind that although prokaryotic cells are pretty simple, they are still highly effective in how they're able to survive in their environments.
In contrast, eukaryotes are much more complex. And unlike prokaryotic cells, eukaryotic cells are capable of coming together to form multicellular organisms, like you and me, or the trees around us, your pets, so many forms of life. Starting from the outside and working our way in, similar to prokaryotes, eukaryotic cells also have a protective barrier. For some, that is simply the plasma membrane, such as what we see in our animal cells, while for others, that is a plasma membrane as well as a cell wall, such as that seen in plant cells. Within the plasma membrane is cytoplasm, and that cytoplasm, which we touched on just briefly with our prokaryotic friends, that is a jelly-like substance where everything inside of the cell resides. For clarification, because sometimes people will use the term cytoplasm and cytosol interchangeably, when we are talking about cytoplasm, the cytoplasm is the space within the cell, right? So filling the cytoplasmic space is a jelly-like matrix, and that is called the cytosol. And the cytosol, that jelly-like matrix, is an aqueous or water-containing solution that contains many soluble ions, small molecules, and macromolecules. Exclusive to eukaryotic cells, though, the cytoplasm includes numerous membrane-bound organelles that are suspended in the cytosol, as well as the cytoskeleton, which is a network of fibers which gives cells their shape and also helps with cell motility and transport. The cytoplasm, though, is also where many non-organelle cellular functions can take place, such as protein synthesis from free-floating ribosomes. Furthermore, the aqueous or water-containing nature of the cytoplasm facilitates protein folding by our hydrophobic exclusion that we learned about earlier, right? By repelling hydrophobic amino acid side groups into the protein core, it helps facilitate that proper protein folding. Eukaryotes, like prokaryotes, also have ribosomes and DNA. However, unlike prokaryotes, where DNA is just in a central region of the cell, in eukaryotes, the DNA is enclosed in a membrane-bound organelle called the nucleus. For clarification, let's go ahead and break down what I mean when I say membrane-bound organelle. In your body, you have many organs, right? Such as your stomach, your liver, kidneys, right? And all of these have very specific functions in your body. Well, an organelle simply translates to tiny organ. Kinda cute, right? They are basically the organs of the cell, each with their own specific function. And when I say membrane-bound, that simply refers to an organelle that has a protective membrane or barrier surrounding it that separates it from other components in the cell. Make sense? So DNA in a eukaryotic cell is found in a membrane-bound organelle called the nucleus. Eukaryotic cells actually contain numerous internal compartments, or membrane-bound organelles. These organelles that are membrane-bound allow for very specific functions to occur within microenvironments, protected from both the cytoplasmic interior and the extracellular, meaning the space outside of the cell, extracellular surroundings. For instance, the nucleus requires controlled conditions for precise regulation of the genes or DNA that is inside of the nucleus. Therefore, it's surrounded by a double membrane, the nuclear envelope. And the nuclear envelope has pores or tiny holes in it to control what molecules can enter and leave the nucleus, such as mRNAs. Furthermore, some proteins require an oxidative environment and must be isolated from the cytosol, which is reductive. Therefore, ribosomes produce these proteins within the endoplasmic reticulum, where they can undergo critical modifications. Because the endoplasmic reticulum, since it has that membrane, it is able to facilitate that oxidative environment that's required. Subsequent movements even happen within small chemical microcosms that we call vesicles to continue to protect things from the cytoplasmic space if it is not conducive for them to be there. And then the vesicles will transport materials from one area to the next, such as from the endoplasmic reticulum to the Golgi apparatus. Other organelles, such as our peroxisomes, provide protection from their own damaging byproducts, right? Peroxisomes can produce things that are damaging, such as hydrogen peroxide. But by sequestering enzymes that convert that toxic compound into a harmless molecule, such as water, there is an advantage to the cell, right? So peroxisomes have a membrane surrounding them for protective purposes. Similarly, lysosomes, which kind of function as, you can think of like a stomach in the cell as they digest things, lysosomes segregate enzymes to safeguard digestive reactions that require a much lower pH than the cytoplasm has. Mitochondria are another example. Mitochondria also have acidic interiors between their two membranes. Overall, eukaryotic compartmentalization supports actions and processes that would otherwise not be possible if they were carried out simultaneously without some form of containment. And this is why eukaryotic cells have such an advantage to having these membrane-bound organelles within them.